Each nucleotide has a nitrogen-like sugar called deoxyribose in the phosphate group. Phosphate backbones to the structure that is the structure that helps and makes the DNA. DNA has genes and chromosomes. The 4 types of DNA are adenine, guanine, thymine, and cytosine. Thymine is always with adenine, and guanine is always with cytosine. They are very important parts of the DNA, and they never change on each other. DNA functions your cells. It has a blueprint, which is the directions to control your other genes, such as proteins and RNA cells. I use Wikipedia. I got this from you guys, learning different information, and I wrote it down. And I got most of my pictures from Google Images, and those were all the websites that I used. So yeah, I'm finished. Hope you enjoyed my project. Okay, these are the phosphate sugars and deoxyribose. The lines are the sugars, and the circles are the phosphates. And the lines in the middle with the dot are the deoxyribose, which makes the phosphates and the sugars. And DNA also makes us human traits and genes. Also, these are the helicase DNAs and the polymerase. These are the helicases and the polymerase of the DNA. The DNA polymerase inserts the nucleotides from the bonds and proofreads. That's what all the boxes are for. The helicase uncoils and separates the DNA from each other, and the ligase bonds the fragments together. This is the cell cycle. For growth 1, the cells grow organelles and they duplicate each other. The synthesis chromosomes duplicate from 46 to 92 chromosomes. It has a checkpoint. The growth makes corrections for the DNA, and the mitosis makes all the cells divide. Growth 1 has 46 chromosomes, and the synthesis has 92 chromosomes. The body cells are red blood cells and white blood cells. The muscle, I'm gonna have to read that. The replication becomes on anti-parallel strands of DNA. The process starts with a short strand of DNA that binds by pairing its nucleotide. Bases to those in the DNA strand can be replicated in an exposed sugar molecule at its end. So the DNA is together, but then it splits apart because of the process with the nucleotides together makes the DNA split apart. The DNA has 2 strands now, and they are completely separated apart from each other. The mRNA comes from the DNA to the ribosome, and the ribosome and the tRNA connects with the ribosome, and they both connect together to make amino acids, which will become a protein. The translation process incorporates 20 different amino acids, and the precise sequence dictated by 3 base codes built from the alphabet of 4 bases. The process in the ribosome builds the polypeptide chains that will become proteins. The RNA connects with the DNA, and they connect together all the bases, and it comes out to the cytoplasm. It has the coding strand, the template strand, the RNA DNA, the rDNA, and RNA hybrid region. And the nucleotide was being added to the third end of the RNA. Point-shift mutation is a mutation that causes a single nucleotide base change, insertion, or deletion of genetic material.
I want to make a teaching parody of what
caused by insertions or deletions of a number of nucleotides.
In which the kids are like raising their hand and looking at the teacher and being like, what's that? And then the teacher goes over to them and she's like, running out of time. And the kid's like, what do you need? You need to make up your mind.
I got my stuff from Wikipedia, information from you guys, and most of my pictures from Google Images. And that's it. Hope you enjoyed my project.